Hello, everyone. My name is Harriet Kluger. I'm a professor of medicine and medical oncology at Yale University. On the clinical side of my job, I take care of patients with melanoma and kidney cancer. And in this capacity, I had the privilege of meeting and taking care of Rob towards the end of his life. So I just want to take 1 minute to just describe our interaction with Rob and how hard he fought to win this battle. He came to us a few months before he passed, and he was really motivated to succeed and to go on a very intense clinical trial. And during this period, we got to know him and his family very well. And I have to say, we just loved Rob and Joan so much that we really all thought that we should move down from New Haven to New Jersey, because obviously everyone in New Jersey is so amazing. But on behalf of Rob, I'd like to say that the fight's not over, and I'm sure he would have been thrilled to see each and every one of you out here today trying to keep this mission going so that future patients will do well with improved therapies for metastatic melanoma. So a number of years ago, when I started treating patients with metastatic melanoma, which means melanoma that's spread beyond the initial site to an internal organ, we would sit the person down and say, I'm sorry, Mr. Jones or Mrs. Jones, on average, you have approximately 6 months to live. You should get your affairs in order. A few years later, in around 2004, 2005, when the newer immune therapies that we're now giving were coming in through the clinical trials, we noted that our patients were actually living longer, and we modified our discussion to say 9 months to a year. At this point now, in 2017, we actually don't have that conversation anymore, because at least 50–60% of our patients are living for a long time. We don't know how to define a long time yet, because these therapies are relatively new, but we're hoping that they might actually live forever. The problem, though, is that it's not 100% of our patients, and I think that the fight is still on. We need to get to the point where 100% of our patients are cured from this awful disease. So the major breakthroughs in treating metastatic melanoma really started with treating a certain kind of immune cell called T cells. So when we can activate these T cells, we make them fight against the cancer, as opposed to giving something like chemotherapy, where we try to actually target the cancer cell itself. Now, it turns out that these immune therapies that target T cells are good in a certain percentage of patients, as I just said. But one of the reasons that they're not good in all patients is that there's a second type of immune cell sitting in the tumor called macrophage. Now, there are various types of macrophages. At least 2 are well characterized within cancer cells. And some of them promote tumors, and some of them actually inhibit tumors. So I think that the next generation of immune therapies is going to try to target or manipulate these macrophages to turn them into tumor-inhibiting macrophages as opposed to tumor-enhancing macrophages. And this is what we're proposing to do with the funds that you've raised today. So more specifically, we're going to start with collecting samples from patients who are treated with these T-cell targeting therapies, and characterize their macrophages from both their tumor and from their blood to see whether the patients who do not respond to these T-cell enhancing therapies have more or less of one or other type of macrophages. Then our second goal is to take mouse models that we have. These are immune competent mice with intact immune systems. Some of them respond to these types of therapies and some of them don't. And we would then like to characterize the macrophages in the tumors within the mice and compare them to the human tumors. And if we go back and forth between the patient samples and the mouse samples, we'll be able to determine exactly how we should manipulate these macrophages in order to make our T-cell targeting therapies work better. So this is going to take us a few years, but we're proposing these first steps in the next 12 months. And I just want to thank all of you for your contributions today, because this will help us move this field forward. And I also hope you all had a good golf game and enjoy the rest of your day.
I want to thank the Stoffel family and all the organizers of Golf for Rob and for your generous donation to melanoma research. I really wish I was there to thank you in person, because of all the awards and grants I've ever received, this is the most meaningful. This is because Rob exemplifies why I do translational research, to take what I learned from the clinic to the basic science lab and back to the clinic with the hope of developing new and better therapies. 4 years ago, I came to Penn to study cancer and to do a cancer fellowship. I joined a research program co-mentored by Tharg and Gowder on a clinical side, and John Wary, a T-cell immunologist, on a basic science side. And our goal was to study T-cell dysfunction in melanoma and how it can be reversed, whether we could use antibodies that target the molecule PD-1. And although he didn't know it, Rob played a role in my research career. Through a collaboration between our lab and the melanoma research program at Penn, we published a paper that came out in Nature a month ago. Rob donated blood for this study and played a role in it. In the study, we described in patients with melanoma who've been treated with PD-1 blockade that the balance between their immune system reinvigoration and their overall tumor burden played a large role in dictating whether they had a clinical response and benefit. For a subset of these patients, they had stable disease, which means that their tumors neither shrank or grew to a significant degree on therapy, at least for a time. And for these patients, we could see that their reinvigoration of their immune system and their overall tumor burden was at a perfect balance, suggesting a stalemate in the fight against cancer. Rob was one of these patients. And I remember Thor and I sitting down in clinic discussing this issue with him, that he had a tumor in his lymph node on his shoulder that was stable while he was on pembrolizumab, but now is slowly starting to grow. We actually ended up getting a biopsy of that tumor, and indeed there were tumor cells there, but also there was a lot of immune cells there. This was consistent with the idea that his immune system had fought the tumor to a stalemate up to this point, but now is slowly starting to give way. And this highlights the limitation of our research now and currently is that while we were able to find an important observation, we still weren't able to intervene and come up with a therapy that was ultimately successful for Rob. Now, while we were able to identify the call for reinforcements from his immune system, we weren't able to provide the resources necessary to win the war. And this is where I plan to devote the resources that you have given me through this grant to melanoma research. Thor and I have set up a clinical trial where we're able to collect samples in terms of blood and tumor, both pre-treatment and after treatment from patients with melanoma on anti-PD-1 therapy. And our goal is to study the immune response generated by PD-1 blockade and how it's different between patients who have clinical benefit and those who don't. Moreover, we want to study how the immune system changes after the initiation of drugs to see whether it can give us ideas and novel targets on how to have new and better therapies. Essentially, our hope is that through this grant, we can take steps in one, to essentially understand why these immune cells, while they were in Rob's tumor, could not eliminate his cancer. And two, to provide new treatments in order to tip the balance so that the immune system can ultimately win the war. Patients like Rob is the reason why it's a privilege to do what I do. The need for new cancer therapies is so great, and there's so many important questions to answer in science. But ultimately, it's actually seeing patients like Rob in the clinic that makes my research meaningful and worthwhile. So finally, I want to end by thanking Rob for who he was, and his dedication and his commitment to melanoma research here at Penn, and thank all of you.